So let me introduce myself first. So I'm Carlos Rewell. I'm in the microbiology and immunology department at the graduate school. And we work with Streptococcus pneumonia. And about 4 years ago, we started looking at how disease is different in the context of an aged animal versus a young animal. Most of the research, most of the basic biology that's done looking at how pneumococcus causes disease is actually done using, it's in the context of disease in children, in very young children and very young animals, laboratory animals, in part because there's a tremendous disease burden in children. And people forget that the majority of the mortality is actually in the elderly, or at least it's not given the same amount of priority. So we started to investigate this, and Pooja was a postdoc in my laboratory at the time, and so the project sort of fell on her, and we discovered rather interestingly that, I don't want to steal her thunder, so I don't say too much, that disease was accelerated in the elderly for some very good reasons that had to do with inflammation. And then Pooja went on to help describe what was one of the causes for this inflammation in the aged animals. And we think that's very relevant to a clinical setting in regards to humans. And importantly, I think maybe at the end of her talk she'll suggest that maybe countering some of this inflammation with drugs or with exercise or so forth is possibly a way to protect against community-acquired pneumonia. So there's a potential therapeutic here. So with that, Pooja.
Thank you, Dr. Rewell, and thank you all for coming. My name is Pooja Shivshankar, and today I'm going to talk to you about my study on age-associated susceptibility to pneumonia. And this was done in microbiology and immunology department. Now I'm with Dr. Claude in medicine, cardiology, and I'm carrying out the same work with regard to idiopathic pulmonary fibrosis. So it's interlinked, and it is the same thing which I'm continuing. So if I give you all like 10 seconds to define aging, we will all come up with several different answers. And those all will be correct, but aging biologists put it in a very simple manner. They say it is a progressive decline in multiple organs function due to oxidative stress, DNA damage, chronic inflammation, the three main aspects that I'll be covering in my talk. And besides, we have mitochondrial dysfunction and hormonal imbalance. As Dr. Ariwala was saying, pneumonia in the elderly is a life-threatening disease, and it affects the lungs, and the causative factors are gram-positive bacteria such as Streptococcus pneumoniae, Staph aureus, and group A Streptococcus, and even gram-negative bacteria, which is Haemophilus influenza, and viruses such as influenza primarily, and human parainfluenza virus, as well as respiratory syncytial virus. RSV was known to be a typical infant pathogen, but it is gaining more and more importance in the elderly with respect to community-acquired pneumonia. So whatsoever the cause of the community-acquired pneumonia in the elderly, the percentage with Streptococcus pneumoniae is more and significant worldwide compared to other pathogens. So the treatments we have are a vaccine, which is Pneumovax 23, which has to be given on yearly basis because the elderly lack robust immune response. So we have to keep them boosting periodically. And once infection is established, we have antibiotics such as beta-lactams, macrolides, and fluoroquinolones. And always a combination therapy proves beneficial in these patients. The reason is many of these Streptococcus strains have developed resistance towards beta-lactam. So with all those treatments and improvements, again, the question remains for effectiveness of these treatments. And that's the reason we want to know what else is responsible besides aging, because aging is inevitable. We think in our mind the first two things to be the causative factor is chronic obstructive pulmonary disease and idiopathic pulmonary fibrosis. COPD is the fourth leading cause of death in the United States, and by 2020 it's going to be the third leading cause of death. And so the annual incidence of IPF is like 30,000, and 73% of those population, the patients are in the age group of 65 and above. I don't know why the graph did not display it. And not only those two age-associated lung pathologies, we have systemic inflammation as such as a major risk factor for the incidence of community-acquired pneumonia in the elderly. And et al. Has shown that comorbid conditions such as cardiovascular diseases, diabetes, and smoking habits, oxidative stress basically, can all increase systemic levels of pro-inflammatory cytokines such as interleukin-6 and TNF-alpha, which are the major risk factors associated with the incidence of community-acquired pneumonia. In the same line, Dr. Ariola has shown that age-associated inflammation using animal models increases susceptibility to pneumococcal pneumonia, and the mechanism behind that was an increased levels of two receptors basically that are used by Streptococcus pneumoniae, PIGR and PAF receptor, and they are held to get invaded. And we wanted to mimic the condition of aged mice into young mice, and to do that, we infused TNF-alpha. We generally raised the levels of circulatory TNF-alpha and showed that indeed there is an increase in PIGR and PAF receptor levels, and this was because of NF-kappa B activation, which is upregulated by the inflammatory cytokines. And these two proteins are controlled by the NF-kappa B. Therefore, we also saw an increase in the bacterial burden in the lungs as well as blood in TNF-alpha infused young animals, which are mimicking like aged animals compared to the previous controls. So with that, the model we proposed was, in aging, you have increased chronic inflammation that upregulates these receptors so that they prime the lungs for increased bacterial bindings. And once the infection is established, you have a delayed recovery phase because of poor innate immune response due to toll-like receptor dysfunction compared to the young animals. So, age-associated inflammation is now responsible for the upregulation of these receptors and recruitment on the cell surface, and it could be extrapolated to the situation which might be faced by IPF and COPD patients who are vulnerable to microbial assaults. We went on to identify two more ligands which are opted by the pneumococcus. And keratin 10 was a very unique host receptor for the bacterial adhesin PSRP. And I want to point this out because I'll be covering in the age-associated manner these receptors also. And Dr. Aruela has shown that laminin receptor facilitates bacterial binding on the meningococcus. Those are endothelial cells. And so once you have bacteremia in aged patients, it does not delay the meningitis stage. So that's the major risk in the elderly. So when we were looking for the aging samples, the same samples that we used for the analysis of PIGR and PAF receptor, we also recruited human tissue samples. And these human tissue samples are obtained from Lung Tissue Research Consortium. And they're basically from the patients who undergo surgical resection for lung cancer and COPD. And it is a distal portion of the lung which is considered as normal from the pathologic side. And we categorize them into different age groups. And what we are seeing is an increased level of keratin 10 in an age-dependent manner, and laminin receptor as well as PAF receptor. We use PAF receptor as control for this study. And we do have an increase definitely in mice lungs, and laminin receptor is also elevated. Although we had laminin receptor seen in the endothelial cells, Dr. Aruela was very curious to know whether laminin receptor is also present on lung cells. And for our luck, we found that laminin receptor was present on the lung cells too. And the receptors are very peculiar. Colon binding protein is the pneumococcal adhesin that is that uses both laminin receptor as well as PIGR receptor, and PAF receptor is used by the cell wall component, which is phosphorylcholine. And as I told you, PSRP takes up keratin 10. And what you're seeing in the aged situation is like 10 times to 20 times more receptors expressed on those lung cell surface compared to the young ones, facilitating bacterial adhesion as well as dissemination. And more importantly, keratin 10 is used by other two serious pathogens which affect the elderly. One is Streptococcus aureus and Bruteus streptococcus. And likewise, laminin receptor and PAF receptor are used by Haemophilus influenzae and Neisseria meningitidis. So this is a big concern. So what can we do so that we can reduce this mystery, resolve this mystery and lead towards healthy living? We have to change the perspective for life. And to find a link between inflammation, IPF, COPD, and the incidence of community-acquired pneumonia, we went through the literature. And the clue we got was senescence. In COPD patients, it is reported that those emphysematous lesions have senescent cells, and those are type 2 pneumocytes, and their NF-kappa being activated. And we were also lucky to find a suggestion given by Verma et al. That senescence could be a phenotype in the onset of idiopathic pulmonary fibrosis. So we know that there is something going on with the cell property. So what is cellular senescence? Senescence is a phenomenon where cells lose the capacity to replicate. Normally, a cell can divide up to 60 doubling times, and after that, they retire. It's called Hayflick limit. So if they retire, they just sit there and they're metabolically active, and they alter the physiological function of the normal neighboring cells. Judith Campisi, she's the pioneer of cellular senescence studies, and she has shown in the context of tumor progression that there are two pathways, signaling pathways, which lead towards senescence. One is considered as a reversible pathway, and the other one is irreversible pathway, which is irreversible pathway is the one which is more predominant in aging, normal aging. So the P53 and P21 pathway leads to a reversible senescence, where the cells can revert and They can even get transformed as malignant tumor, or they can become normal. And in irreversible pathway, they just have to wait until the programmed cell death occurs. And the one negative consequence of these senescent cells was shown by, again, Geriatrics, that they secrete increased amount of pro-inflammatory cytokines and growth factors and proteases, which dysregulate the function of the normal cells. And this paracrine effect can either attract the chemotactic factors to clear the tumor cells, or it can induce tumor progression through angiogenesis and metastasis. So now we know that something is going on with the aging cells. To develop this hypothesis, and we went ahead to study cellular senescence as a factor to contribute to age-associated inflammation and as a factor for increased pneumococcal infection. So, to prove that, we first had to show that senescence does occur in aged lungs. And we used the same aged mice and aged human lung samples and studied the pathway of P16 and PRB. As you're seeing here, there's a significant increase in the mature and aged groups of human lung tissues in the P16 levels and PRB levels, and a significant increase in the ultimate heterochromatin regulatory protein, which is macrohistone 2A. And we also see a discrete increase in the levels of P16 and PRB in the mouse lungs. So this suggested that cellular senescence does occur during normal healthy aging. So are we going to prove that with chronic inflammation? Yes, there was a link between increased cytokines in those lung tissues, and we also confirmed that with pathology, showing that there is increased inflammatory cytokines, inflammatory cells in the aged tissues. And since we know that K10 and PSRP interaction is very specific for lungs, we wanted to show that aged mice susceptibility to the pneumococcal infection could be dependent on this vital K10 PSRP interaction. So I don't want to confuse you, but what we are seeing here is aged mice are susceptible to pneumococcal challenge compared to the young ones, and the infection was attenuated when we used bacteria which was deficient in PSRP, suggesting that PSRP K10 interaction was very much necessary, and K10 upregulation is significant to establish the infection in the lungs. And not only in the lungs, the dissemination also was faster in the aged animals compared to the young animals, as we see in the bacterial burden. So now that we know the aging increases senescence, aging is associated with chronic inflammation, aging has shown susceptibility to increased bacterial infection, how are we going to prove that it is due to senescence? To prove that, we had to use an in vitro study. For that, we used lung cells, which are pneumocytes, and we used bleomycin. It's an anti-cancer drug, and it has been used by many investigators to induce senescence. So there is a standard senescence marker, which is senescence-associated beta-gal lactose-base activity, and this is an acridine orange and ethidium bromide stain. Basically, it shows that if the dye is not taken up, the cells remain green, which are metabolically active, and if this dye is taken up, they turn yellow and they are dead. So what we are seeing is they are senescent and they are metabolically active, and there is an increase in the levels of keratin 10 and laminin receptor in a dose-dependent manner, whereas PAF receptor levels didn't change. That was surprising. And then indeed there is an increase in the markers P16 and triple PRB and macro-H2A, which correlated very well with the increased bacterial binding of those cells, suggesting that senescent cells do increase permissiveness of these bacterial binding on the cells. And how about the paracrine effect? To know the paracrine effect, what we did is we used the conditioned media from those senescent cells, and we incubated normal fresh cultures with that media for 2 hours. And when incubated, it is just like how we can incubate normal cells with inflammatory cytokines like recombinant TNF-alpha and IL-6 to trigger inflammatory response, and even apoptosis we can see in that. So senescent media, conditioned media, increased the levels of IL-1-alpha, IL-6 and IL-8, and there was increased permissiveness to the bacteria on those fresh cells exclusively through PAF receptor levels, which was really interesting because PAF receptor is activated through NF-kappa B activation, and these inflammatory mediators, you can simulate the same condition in vivo, that these inflammatory mediators can go and trigger normal cells, and what you're seeing is majoritively PAF receptor expression, and that's more than enough because it's a ubiquitous protein and it can help the bacteria to bind to, attach to, and invade and disseminate into the bloodstream. So, since we now know that senescence is associated with increased bacterial binding, how can we relate to the situation which the IPF patients and COPD patients face? If it is going to be correlated with those disease pathologies, we had to mimic those conditions in animal model. So what we did is, again, bleomycin is an anti-cancer drug which has been used by many investigators to induce pulmonary fibrosis, because bleomycin's side effect is pulmonary fibrosis. So using bleomycin, we induce pulmonary fibrosis in young animals, and for oxidative stress, we used hydrogen peroxide, because in COPD patients, what we see in majority is generalized oxidative stress. So what you're seeing here is, again, the same markers. PRB, P16 is discretely increased, and there is an increased amount of laminin receptor and PAF receptor. We did not see any change in creatine 10 levels. So anyways, we have now 2 receptors which are upregulated in the conditions of IPF and COPD. And we have the senescence markers, definitely the P16 and PRB. Many of the studies which are done on pulmonary fibrosis, they show P53 and P21 pathways predominantly. And we are the first one to show P16 and PRB pathway. And bacterial burden, of course, was very high in these animals when compared to the placebo controls. So this suggested that COPD patients as well as IPF patients may have senescent cells which might entertain these bacterial pathogens to bind to and cause acute exacerbation of these disorders. That's the reason even these patients, when they are given vaccination, for example, they have acute exacerbation of the disease, so we have to be on check to see how they react to the situation. And of course, the inflammatory markers were very high, and the disease pathology, as you see in the histological section, matched to the human pathologies. It's a fibrotic lung with a lot of inflammation, and we see an emphysematous lesion when we gave hydrogen peroxide to these animals. And this suggested a model where we showed not only smoking or DNA damage, which are the causative factors of COPD and IPF pathology, even normal aging can induce cellular senescence, which increases the bacterial ligands, PAF receptor, K10, and laminin receptor, along with increased inflammation. And those pro-inflammatory mediators, which are released from senescent cells, can affect the normal cells and at least increase one of the receptors, which are very important and for bacterial binding, which is PAF receptor, and increase even inflammation in those cells. So basically, we are triggering or we are dysregulating the property of normal cells also The options, this I don't know, but I read to the literature. So any comorbidities like cardiovascular diseases, diabetes, they are common even in patients with IPF and COPD. And they're advised to be on oxygen therapy because they have difficulty in breathing. They're recommended to take vaccination against pneumococcal infection as well as influenza. And statins are very good drug which are prescribed for cardiovascular disease patients. And we have substantial data shown by Dr. Ariela's group that statins protect against pneumococcal infections in the aged mice. And he'll be presenting that maybe in his next talk. And there is one more drug which is on phase 3 clinical trial, which is IL-1 receptor antagonist. It is now, I don't know whether it is FDA approved or not, but still it is given to most of the arthritis patients to curtail chronic inflammation. And last but not least, social life and healthy living along with exercise proves beneficial in preventing community-acquired pneumonia in these elderly patients. And the overall summary, we know now that senescence is there in aged lungs, and it increases through sacs, the pneumococcal ligand expression on the host cell surface, and anti-inflammatory and immunomodulatory drugs can prove beneficial for patients to prevent them from microbial assaults. And with that, I would like to acknowledge Dr. Ariela for giving me the freedom to work on this topic, and Dr. Lisa, who is our collaborator and my current boss, and the grants from NIH and Leah Foundation, and San Antonio Greg for giving me the opportunity to talk. Thank you.
Anybody have any questions?
Pooja, I walked in late, but what I could ask you, you said Patrick receptor?
Yeah.
Are there any knockouts of that or anything?
Yes, that receptor knockouts are available.
And they are fine?
Yeah, they're fine. Dr. Ariela has done work on PAF
We don't
receptor.
know, because you would need 2-year-old PAF receptors.
Right, that's what I was,
So people have done work with young PAF receptor knockout mice, and they're generally a little bit less susceptible, but no one's looked at 2-year-old.
So any work with that would be like, would be new, right? And
Yes.
the aged back.
Yeah, it's difficult to get. That's actually a very difficult term to do because you would have to breed those mice and then keep them for 2 years. So you'd have to have a very large colony for 2 years. Each of those mice that we buy is about $250 each. So an experiment with 12 mice is $3,000. So that's.
And I have 2-year-old aged beta-2 adrenergic receptor knockout mice, and my costs are horrendous, so yes, I
Right.
understand it.
What did you see the Pneumovax recommendation?
Pneumovax 23.
Yeah, how often?
I'm sorry.
I thought you'd say how often it's given.
It's recommended to be given yearly,
No,
5 years.
5 years, but yeah, I just
Okay.
wanted to clear, yeah, okay. All right.
I have a question. So I'm just a medical student. Could you explain the relationship between aging and why it's assumed that inflammation does occur, the research behind that?
Because aging increases oxidative stress, so generally there are so many reactive oxygen species, and by time you have telomere dysfunction, DNA damage, so that increases inflammation in your body. So it's chronic inflammation which is there in your body, and you're carrying that through your whole life as you age.
Okay.
So, and mostly that's how they get triggered to arthritis and other diabetes or inflammatory diseases, and even obesity.
So this is a very good question because one of the questions is, so there is chronic inflammation in the aging. Now it's nowhere near the levels during like an infection. This infection would be way up here, a young person's way down here. chronic age-associated inflammation is near the bottom, but it's elevated. There's questions as to why that's there, and it's really multi-factorial. It could be because you're overweight, you have diabetes, it could be because you are smoked for 30 years of your life, it could be because you worked at a plant and you inhaled stuff, it could be because you have cancer, it could be, so Alzheimer's, Parkinson's, atherosclerosis. Then there's the normal aging biology processes like oxidative stress, senescence. All of these are happening at the same time. And so the causes of inflammation are very varied, but it's generally accepted that the older you get, the more TNF-alpha and interleukin-6 C-reactive protein that you have in your blood.
Okay.
And so, and there's a marker for inflammation called NF-kappa B. If they take tissues from aged animals and aged humans, they see that NF-kappa B is activated in these aged tissues. So it's not a lot of inflammation, but it's enough to kind of just tweak you towards what we're observing.
Thank you.
Does the amount of inflammation vary depending on what kind of tissue it is, or does all tissue pretty much, the inflammation is the same with all kinds of tissue? Like liver tissue or muscle tissue?
I think people are studying with reactive oxygen species, and the generation of ROS in most of the tissues is more. For example, even arthritis patients, they are having lesions in the chondrocytes, and those chondrocytes undergo senescence, and atherosclerotic lesions, vascular endothelial cells undergo senescence. So wherever you have aging cells, you have inflammation. So all these disorders are linked to those inflammatory conditions. Even this one more, diabetes, pancreatic cells also have inflammation, and Alzheimer's, neurodegeneration is there. So
We've seen
if
a
you
lot
see
in the
multiple
HIV,
organs,
in the HIV
patients what you
a lot of
so,
inflammation.
yeah, so you have inflammation in all the sites. Even strep pneumo has gone up to even arthritic, those cavities. In osteoarthritis, people have seen have extracted strep pneumo cultures from there. So that means that wherever you have endothelial cells, and BAFF receptors are really the culprits, where they take everywhere, and even endothelial cells. If you see BAFF receptor causes the bacterial cell wall components to cross to the atherosclerotic plugs. And Dr. Ariwala has shown that there is cell wall components aggregated in the endothelial cells of the vasculature. So that way, if you see, yes, we are having inflammation.
So one of the questions is, why would having diabetes make you more likely to have pneumonia? Or why would having atherosclerosis make you more likely to have pneumonia? But those are established risk factors for community-acquired pneumonia. And it seems that the common denominator is inflammation. All of those symptoms cause low-grade systemic inflammation. And the study where she used the mice with the TNF-alpha, we put the pumps in, that shows that systemic inflammation is enough to change the expression of proteins within the lungs. So systemic inflammation makes your lungs stickier for bacteria. If you're older, that might be enough to increase the incidence of disease.
Do you think it's the inflammatory conditions that are responsible for the upregulation of keratin? And if what's the normal function of keratin? Why would it be?
Yeah, that's a good
Sorry.
question. Keratin 10 is a terminal differentiation factor for keratinocytes, and it is very lately identified on endothelial cells and lung cells by our group. Endothelial cells, only last year that paper got published with another pathogen. So, and it also induces autoimmunity, keratin 10. So it's a differentiation factor on keratinocytes which controls the cell proliferation from the pathway of PRB, P16 and PRB, and it sequesters AKT phosphorylation. That's the mechanism where it arrests the cell phosphorylation, cell proliferation. So if that is the case, keratin 10 is stimulated through IL-1 alpha, which triggers CEBP beta. CEBP beta is the transcription factor for K10 upregulated. So there is direct relationship between senescence induction, where you have so much of intracellular IL-1 alpha, and which triggers CBP pathway.
And so there's a crosstalk between CBP and NF-kappa being in the cell?
Yes, there are promoters which have two binding sites. For example, laminin receptor has 2 binding sites, AP2 and CVP beta. So anything can go and attack those, promote those transcriptions.
You mentioned the study with the IL-1 receptor antagonist. Are you using Anacaro?
No, we don't. We are not studying right now, but we plan to do that. Yeah.
I'm more along the lines of fish oil.
Okay,
Yeah.
not,
I think
not so
that's
strong.
not so strong.
It seems somewhat counterintuitive that
Yeah, but it's a two-edged sword, right? More you dampen inflammation too much, and now you're going to make the person more susceptible. Statins seem to strike that balance quite well, just incidentally. And there's lots of papers that show, lots of meta-analyses that show. In fact, it's done by Eric Mortensen and Mark Restrepo here at the Bailick, that show that statins protect against immune-acquired pneumonia. And we think that it's polysaturated fat, PUFAs, whatever you want to call it, fish oil, would also do the same thing. Right. IL receptor 1,
Yeah.
that's pretty heavy. That's
I
pretty
wanted
heavy.
to continue that.
That's a pretty heavy hammer to wield.
Yeah.
I'd
It's very expensive.
I'd rather you just
actually.
buy it at the grocery store. Just take it.
So were the statins related to atherosclerosis or across the board for diabetes, anything it helped improve?
No, actually, statins don't help everything. I think they make diabetes worse. I can't remember that, but
Okay.
they do inhibit NF-κB activation. And we have data that shows young and aged mice on statins have lower lung damage, lower disease severity.
Okay.
We actually think statins would be a very good therapeutic for kids who have sickle cell disease, because they have a 500-fold increase in the incidence of pneumococcal pneumonia due to this chronic vascular inflammation. It's very interesting. It's very specific to the pneumococcus, not just encapsulated bacteria, because they don't have a functional spleen. But so they're susceptible to all unencapsulated bacteria, but they're especially susceptible to pneumococcus. So we think that statins are possibly a very good therapeutic for them.
Inflammation is like stepping into everything, because I work with fatty liver disease, but now fatty liver disease is just plain hepatic steatosis was not considered to be benign, but now you're having more and more cardiovascular disease associated with just fat in the liver. That's all, not even NASH or steatohepatitis, and that's all because liver's then releasing CRP and IL-6. And it's going to the heart, so.
So I'm going to take I'm taking fish oil right now. At 250, I'm going to find one of you, get some statins, and we'll see what happens.
And maybe get your pneumonia shot.
I already have it. Well, I work with it, so I get immunized every day.
And even amycin.
So that's it? All right.
Thank you.
Thank you very much.